Hello students, welcome to the channel Physiology Redefined. In my earlier video, I talked about the general outline of the hematopoiesis. That means from the pluripotent stem cells or from the hemocytoblast, which has got the multipotentiality. That means which has got the potentiality to develop into different series of the blood cells, RBC, WBC and the platelets. So all these cells are developed from a single mother cell, that is known as pluripotent stem cell or hemocytoblast. And from the hemocytoblast, all the blood cells are formed, and the general outline I have given in my earlier video. So in this video, I'll talk about the erythropoiesis, process of erythropoiesis. That means how from the hemocytoblast, how from the pluripotential stem cells, the mature RBC is getting formed. So this process of RBC formation from the mother cell, that is hemocytoblast, is known as the erythropoiesis. So in this video, I will talk about the mechanism of the RBC formation or erythropoiesis. So please watch this video till the end, and if you like it, don't forget to share it. Don't forget to press the like button and also subscribe the channel if not yet subscribed. So let us see what is erythropoiesis and how the RBC is getting formed from the hemocytoblast. As I told earlier also, that from the hemocytoblast, two types of stem cells are formed. Those are known as the committed stem cells, and these committed stem cells are of two types. One is known as the myeloid stem cell series, and another one is the lymphoid stem cell series. From the myeloid stem cell series, three categories of the stem lines are developed. One is the CFU of E series that develops into RBC. Another is the CFU of M series that develops into megakaryocytes or platelets. And third one is the CFU of GM series, from which the granulocytes and the monocytes are developed. So in this video, I will specifically talk about the RBC formation. So from the hemocytoblast, how the RBCs are getting formed, that is the topics of discussion in this video. So RBCs are formed in different steps from the hemocytoblast, different stages. So what is the first stage? In the first stage, the pluripotential stem cell, this is converted to the committed stem cells. And which one? This one. So from the hemocytoblast, this committed stem cells is formed, that is the myeloid stem series. Myeloid series. Or in short, myeloid stem series. This is the first. In the first step, the hemocytoblast is developed into the myeloid stem series. So this is the hemocytoblast. That is the pluripotential stem cell. This is developed into the committed stem cells, that is known as the myeloid stem cell. So this is the first stage. In the first stage of the erythropoiesis, from the hemocytoblast, the committed stem cells, that is myeloid stem series, is formed. And from this, in the second step, in the next step, what's happened? The committed stem cell is differentiated to the progenitor cell. So in the second step, committed stem cells, that is myeloid stem cell series, these are differentiated to the progenitor cells. For the progenitor cells, for the RBC formations, that is known as the first is the BFU of E series, and then is the CFU of E series. First is the blast forming unit of the erythrocyte series, and then is the colony forming unit of the erythrocyte series. So this is the second stage of the erythropoiesis. So second stage is the committed stem cell is differentiated into the BFU of E series and thereby CFU of the E series, which are known as the progenitor cell. Now in the third steps, these progenitor cells, these are converted to the precursor cells. So what are the precursor cells? Different levels of the precursor cells are there. So first, this is the CFU of the E series. So in the first precursor cell that is differentiated from the CFU of the E series is known as the pronormoblast. So this is the pronormoblast. So this is the first precursor cell of the stage 3, that is the progenitor cells are differentiated into the precursor cell. First precursor cell is the pronormoblast. This pronormoblast, these are very large cells with a large nucleus and scanty cytoplasm. Cytoplasm is less. Now this pronormoblast is further differentiated to form the second category, second level of the precursor cell, which is known as the early normoblast. So this is the early normoblast. So this is also the second level of the third stage, that is progenitor cell is converted to the precursor cell. First pronormoblast is formed and then early normoblast is formed. So what's happened in the early normoblast cells? The cell size is still large, though comparatively smaller than the pronormoblast. And nucleus is larger, very large nucleus. Almost 80% of the cell is covered by the nucleus. 80% of the cell is covered by the nucleus. So precursor cells, first precursor cell is the pronormoblast. Second one is the early normoblast. So this is A, this is B. Second is the early normoblast. So early normoblast, as I told, very large nucleus, more than 80% of cell, more than 80% of the cell, and scanty cytoplasm. Comparatively, scanty cytoplasm is there. Scanty cytoplasm is there. Within the cytoplasms, there are a high quantity of the RNA and the polyribosomes. So there is a plenty of polyribosomes, RNA, high concentration of the RNA and polyribosomes. As these cytoplasm contains plenty amount of the polyribosomes and high quantity of the RNA. These are basophilic in nature. So these are basophilic in nature and thereby stains blue in color. So this is the about the cytoplasm. As I told, nucleus is 80%, very large nucleus, and nucleus looks like the spokes of the wheel. It resembles as the spokes of the wheel. You can see it resembles as the spokes of the wheel. Spokes of wheel. Nucleus resembles the spokes of the wheel. And these are the polyribosomes. Plenty of polyribosomes, and the RNA, high quantity of the RNA that is present in the cytoplasm, for which it is basophilic in nature. And in this late part of the early normoblast, hemoglobin starts appearing. Here you see the red colored one. This red colored one is the hemoglobin starts appearing. Here hemoglobin starts appearing. So in the early normoblast, the spoke of wheel type of nucleus, and hemoglobin starts appearing. So this happens in case of early normoblast. So this is the pronormoblast. Now this early normoblast is further differentiated to form the intermediate normoblast. So in the third level of the third stage, the early normoblast is further differentiated to the intermediate normoblast. So here you see so C is intermediate normoblast. So early normoblast is differentiated to the intermediate normoblast. So in the intermediate normoblast, you see cell size shrinks, cell size becomes less, nucleus also shrinks, size of the nucleus becomes very, very less, and rather cytoplasm is more. So cytoplasm is more cytoplasms. In the cytoplasm, what's happened? As I told, in the late part of the early normoblast, the hemoglobin starts appearing. So now there will be more synthesis of the hemoglobin. Here you see the red color concentration is much more hemoglobin. So more hemoglobins are synthesized. So in this case, more hemoglobin are synthesized. But in this cytoplasms of the intermediate normoblast, in this stage, still some polyribosomes are there. So there are still some polyribosomes or RNA are there. Polyribosomes and RNA are still there. As both the polyribosomes are there, they are basophilic in nature, and as the hemoglobin is there, which is acidic in nature, eosinophilic in nature, and thereby these will be known as the polychromatophilic erythrocytes. Because here two colors will be available. Blue color because some polyribosomes and RNA are there, which are basophilic, along with the hemoglobins are there, which are eosinophilic or acidic. So this is the more hemoglobin are synthesized, RNA are still there, and thereby these are polychromatic. For this reason these are polychromatophilic erythrocytes. Polychromatophilic erythrocytes. Now what's happened? This intermediate normoblast is further differentiated to form the late normoblast. So this is the late normoblast. Late normoblast. So this is also stage 3 in the late normoblast. So now from the intermediate normoblast, late normoblasts are formed. Late normoblast. In this late normoblast, what's happened? Cell size is decreased, and nucleus becomes pycnotic nucleus, and ultimately disappear from the cell. So initially there is a very small nucleus about to extruded out, and this about to extruded out small nucleus is known as the pycnotic nucleus, which ultimately extruded out of the cell and thereby disappear from the cell. So this is the characteristic features of the late normoblast. And in this case, the more hemoglobin, as more hemoglobin is there, no ribosomes, more hemoglobin, no ribosomes and RNA. So basophilic characteristics is now withdrawn, only the acidophilic characteristics are there, and thereby cell used to become pinkish. This one, late normoblast, used to become pinkish one. So it is also known as the eosinophilic erythroblast. And finally, now what's happened in the next stage? The precursor cell is converted to form the mature RBC. Precursor cell is now converted to the mature RBC. So this is for late normoblast will be now converted to the structure that is known as the reticulocyte. So this is the reticulocytes. So this is the fourth step of the erythropoiesis process, 4 stages of the erythropoiesis process, that is reticulocytes formation from the late normoblast. And these reticulocytes are having a reticulum, a network structure, a net-like structure, reticulum structure. This is known as the reticulum, net-like structure. And these net-like structures are the remnants of the ribosomes and the nucleus. Net-like structures are the remnants of ribosomes and the disintegrated nucleus. Those gives a network-shaped appearance, and that is why these cells is known as the reticulocytes, which are also known as the juvenile RBC. So from the precursor cell, first juvenile RBC or reticulocytes is formed, which are ultimately differentiated to the mature RBC. This is the mature RBC. So this is how the mature RBC is formed from the hemocytoblast via 4 stages: pluripotential stem cells to committed stem cell, committed stem cells to progenitor cell, progenitor cells to precursor cells, and precursor cells to mature RBC via different steps. What's happened? Hemocytoblast to myeloid stem cell series, then BFU of the E series, CFU of the E series, then pronormoblast, early normoblast, intermediate normoblast, late normoblast, reticulocytes, and mature RBC. I hope the concept regarding the mechanism of the erythropoiesis is very clear to you. If you like this one, please share this to your friend circle, press the like button and subscribe this channel. Thank you.